Kia ora everyone, and so welcome to the final of these mini lectures on the three great fungal mutualisms. And what I'm going to do today is, well, in this mini lecture, is to continue to talk about lichens, and then I'll just rapidly go through the learning objectives for this whole section to make sure that we've covered them all. So I'm going to talk about lichens, but we're going to change gear a bit, and we're going to talk about a very surprising result that was found just a few years ago that has kind of revolutionized our understanding of lichens. And so with the material that we've gone through in the course, what you've had previously, you should be in a good position to be able to understand the details of this surprising new discovery. So I will introduce that, and to do so, I'll turn off my video and we'll go through the slides. Okay, so the new discovery that was made is the discovery that actually most lichens have two different fungal partners. So we used to think that they were composed of a fungus and an alga or a cyanobacterium, but now it seems that many lichens are composed of two fungi and an alga or a cyanobacterium. And this was discovered in 2016, and it was described in this paper here that was published in the journal called Science. And the goal of this part of the lichen material is to understand this paper. So you're going to need to read this paper, and the link to it is provided on Canvas. And so you can read this paper and understand what it is showing. And as I said, what this paper, the basic picture of what this paper has found is that many lichens include two different fungal partners. So one of those is an Ascomycete, and that's well known. So the majority of lichen species can consist of an Ascomycete fungus, although there are some that consist of Basidiomycete fungi. But anyway, they found that many Ascomycete lichens not only contain an Ascomycete, they also contain a Basidiomycete, and that Basidiomycete is a Basidiomycete yeast. So yeast are fungi that live in a single cellular rather than a hyphal state, and this was a complete surprise. Nobody had ever suspected this before. And so what I want you to do is to understand, firstly, how they showed that this new fungal partner, the Basidiomycete yeast, is present in many lichens. So you'll need to have a basic understanding of the techniques that were used. So you should be able to describe how they showed this. And then the second objective is to understand why it was difficult to identify before. So why had nobody even suspected that there was a second fungal partner inside of lichens? And so I'm actually not going to talk about this at all in this lecture. This is for you to do in your own time by reading this paper and going through it. What I will do is I will look a little bit at some of the techniques and give you a little picture of the results that they showed, just to help you understand these techniques, some of which may not be familiar to you all. So let's go to slide number 2. And the first technique that I want to talk about is this technique called FISH. And FISH stands for fluorescent in situ hybridization. And on the right is an electron micrograph picture, and then beside that is a fluorescent in situ hybridization picture, and each of these are of a lichen. So this lichen is kind of a tube-shaped thing. So the cortex is around the outside, that medulla that I talked about is in the middle, and here's a fluorescent in situ hybridization picture of that same thing. So FISH is used to detect DNA or RNA in intact organisms. So you can extract DNA and RNA from organisms and analyze it, but this FISH is a way of looking to see where DNA or RNA sequences that you're interested in are within an organism in situ, hence the name, so in place. And this uses probes that are labeled with fluorescent dyes, hence the name fluorescent. And so these dyes enable you to visualize using a microscope where the DNA or RNA of interest is. So it allows you to see where specific DNA or RNA molecules that you're interested in are present within that organism. And what they were doing is using probes that hybridize to RNA, a specific kind of RNA called ribosomal RNA. And each individual species has different sequences of ribosomal RNA. And so they can use probes to detect specific ribosomal RNA from specific fungi so that you can distinguish where fungi from one species are compared to fungi from another species are, for example. And these are the kind of results. So the different colors represent different probes that are attached to different dyes that are hybridizing to different organisms and showing you where different organisms are. And I'll show a couple of movies where we look at that in a little more detail. The second technique that I want to look at is RNA sequencing, or sometimes known as transcriptome sequencing. And in this, you extract RNA from an organism, so you actually remove the RNA, and you convert that RNA to DNA by a process known as reverse transcription, and that DNA is known as cDNA as a result. You can then sequence that cDNA to determine what genes are being expressed or transcribed in that organism and at that time. So the RNA comes from DNA that's being transcribed. You take that RNA, you turn it into DNA. The reason you do that is it allows you to sequence it, and then you sequence that to determine what genes were had been turned on in the organism at that time. And this uses something called next generation sequencing technologies that you may have heard of in some of your papers. And these are basically just a way that allows you to sequence a massive amount of DNA. And what this does is, because you've converted all the RNA to DNA, it allows you effectively to sequence all the RNA that was present in a cell. And so the idea here, which will become important for this paper, is that if there are multiple organisms present in the particular sample, so lichen in this case, then you will extract RNA from all those organisms, and the RNA from all those different organisms will be sequenced. And by looking at the sequences, you can identify which organisms they come from. So that should give you a little bit of background. You can do further. There's a lot of information about both these techniques available on the web. You can just do Google searches for them if you're having trouble to understand that. But hopefully that gives you enough background to be able to understand the paper. What I'm going to do is I'm going to finish off introducing this paper by showing a couple of movies. And the legends on this slide, sorry, on slide number 4 now, the legends on this slide describe what is being shown in those movies. I'm not going to read those out, but I'm going to break out now and attempt to show those movies. Okay, so this is the first movie, and so that's the name of the lichen that they're looking at. And what they're saying here is that the purple stuff represents the Ascomycete hyphae, and it also represents the secondary metabolites that those hyphae make. And the green represents the Basidiomycete yeast that I talk about. So these are the things they discovered. And so you can see these Basidiomycete yeast embedded in the Ascomycete hyphae. And you can see that a little better now. Now they're going to strip away the Ascomycete to just show you the Basidiomycete. So the Basidiomycetes are embedded in those Ascomycete hyphae. And if you flip this thing upside down, you'll be able to see the hyphal structure of those Ascomycetes a bit better on the upside down view. So this is the top of the outside of the lichen, and then that upside down view was the kind of inside going into the medulla. Okay, let's stop that. Okay, and so this is the second movie, and this is using the fluorescent in situ hybridization, the FISH technique that I looked at before. So the coloration is the same. There's one more additional color that's red, and the red represents the photobiont. So this is looking at the lichen, that kind of tubular structure. So the top here is the outside, and then on the inside is the photobiont that you can see it on the bottom there. And so you can see the Ascomycete hyphae and those Basidiomycete east in green. The Basidiomycetes in green are kind of embedded all throughout that top layer of Ascomycetes. Okay. So, so that's the goal for you, is to look at that paper and be able to understand those 2 points that I've got down the bottom of the slide there, and that's all I'm going to talk about this. So the rest of that is for you to do. And then just to finish off this section, I'm going to go through these learning objectives. We went through these at the start. This is slide number 5. And so by the end of these lectures, you should be able to compare and contrast features, structures that characterize arbuscular or endomycorrhizal symbiosis and ectomycorrhizal symbiosis. So I think we've covered that. Critically analyze the evidence suggesting mycorrhizal associations facilitate communication between host plants. We went through a lot of that. List the 4 classes of fungal endophytes and their major characteristics. So we talked about that. And then using examples, discuss how endophytes can be seen as a continuum between pathogenicity and mutualism. And we use the example of class 1 endophytes to look at that. Using the example of endophyte production of secondary metabolites, explain the ability of endophytes to be maintained by vertical transmission in the host, despite the cost they impose on the host. So we talked about that in terms of the vertical transmission and the benefits that these fungi produce to outweigh the cost they impose. Critically analyze the evidence suggesting endophytes provide protection to their host against fungal pathogens. And so again, we've looked at interpretation of experimental data for that in quite some detail. Describe the composition and structure of lichens, including the roles of the fungal and photosynthetic partners. And so we've done that in the previous mini lecture. Then finally, discuss the evidence suggesting many lichens have 2 different fungal partners, and that's what we talked about today, and you'll be able to do by looking at that paper. So that's it for this section. I'll finish up there. Again, if you have any questions, please direct to Piazza, and Marge is going to take over from me, but I'll come back again for one more lecture looking at phylogeny and population structure of fungal pathogen outbreaks.